Hi everyone. In this video we'll look at the structure of the Pacinian Corpuscle, which is a receptor found in the skin, and then go on to consider what happens when it's stimulated. The Pacinian Corpuscle is shown in the diagram. It's sensitive to pressure and therefore we call it a mechanoreceptor. It consists of a sensory neuron ending surrounded by layers of connective tissue known as lamellae. The layers are separated with a gel between them. The membrane of the sensory neuron ending contains a type of sodium ion channel, which are stretch-mediated. This means that their opening and closing is dependent on a mechanical stimulus, such as stretching or pressure. In the absence of a pressure stimulus, the membrane is polarized. In the last video on resting potential, we saw that this means that the inside of the membrane is negatively charged compared with the outside, and this is due to the unequal distribution of ions across the membrane. In particular, there's a high concentration of sodium ions outside of the neuron axon. When the Pacinian corpuscle is stimulated, in other words, when pressure is applied, the lamellae and membrane become distorted, causing the stretch-mediated sodium ion channels to open. Now sodium ions can diffuse into the axon down their concentration gradient. As they carry a positive charge, this makes the inside of the axon less negative. We say the membrane is depolarized. If the stimulus is weak, in this case if only light pressure is applied, very few stretch-mediated sodium ion channels will open and the degree of depolarization will be very small. The sodium ions will be actively transported out again by the sodium-potassium pump and resting potential is re-established. A slightly firmer pressure would open more stretch-mediated sodium ion channels, leading to a greater degree of depolarization. We can see this on the graph shown by the yellow lines. We call these generator potentials, and unless the depolarization is sufficient enough, the sodium ions are pumped back out and no action potential is initiated within the axon. For an action potential to be produced in the axon, the stimulus must be sufficient to cause enough depolarization to reach -55 millivolts. At this point, referred to as threshold, a different type of sodium ion channel opens. These are voltage-gated sodium ion channels, and their opening leads to a huge influx of sodium ions into the axon, causing massive depolarization. This is an action potential. In the next video, we'll look at action potentials in more detail.